Good morning, good evening, students. My name is J.V. Satyanarayana, and I came from Team Academy. I'm a biology lecturer, and I want to teach today molecular basis of inheritance. Right? Molecular basis of molecular basis of inheritance. So, in this molecular basis of inheritance, we can discuss about DNA structure. So, DNA structure, we can discuss DNA structure. Right? Main. So, DNA is also called as polynucleotide strand. So, in the molecular basis of inheritance, what we will study? In previous subject, you might have studied in principles of inheritance. In principles of inheritance, you might have studied principles of inheritance, how the characters are inherited from one parents to another parent. Sorry, one parent to children, how it will be inherited, you might have studied. So, what is the basis for this? How the characters will transfer from parents to offsprings. This we are going to study in this topic, right? So, totally in this lesson, we are going to discuss about DNA structure. So DNA structure is, so first DNA structure, DNA or structure also called DNA is the DNA is the genetic material in higher organisms. So genetic material. So this genetic material. So earlier, first Mendel genetic material is the basis for this, right? The structure of, so DNA is nothing but, for example, if you take a DNA, so this is DNA. So then this DNA and what is the gene? Let us see the difference between DNA and gene. If you see, this portion is called as, for example, this is total DNA molecule, and in this, if it is can produce code for a protein, then if it is can code for a protein, so then this part of the DNA, this entire DNA molecule, but this portion of DNA can put code for a protein, so that this can be considered as gene. This can be considered as gene. So this is DNA. This is DNA, but this is also DNA. This is also DNA. But this portion of DNA we cannot call it as a gene. So the portion of DNA or the segment of DNA which can produce, which can code for a gene, which can code for a protein is considered as gene, right? So then in the case of Mendel, so already Mendel discussed already. So what is that? He has used the term factors for the genes. He has used what? Factors. So in these factors, so factors are nothing but DNA molecule genes, what we will call now. So these genes are the part of DNA. So DNA and RNA, if you see, what is primitive genetic material? The primitive genetic material is, the primitive genetic material. Which one is primitive genetic material? RNA. RNA is the primitive genetic material. Primitive. So first formed genetic material, RNA. So what are the characters of RNA? RNA is undergoes very fast. RNA undergoes very faster mutation. RNA undergoes very faster mutation. RNA undergoes very faster mutation, right? And RNA acts as RNA acts as RNA acts as the which one? A catalyst. RNA acts as a catalyst. RNA acts as a catalyst, and RNA act as so adapter molecule. RNA act as adapter molecule. RNA acts as an adapter molecule. So RNA undergoes very faster mutation, and RNA acts as a catalyst. RNA is adapter, and RNA has less stability. RNA has less stability. So then, in principles of inheritance, you have studied how characters will transfer from one parent to another parent. So how the characters are transferred, you have studied. And for this characters, what is the base you are going to study in this lesson? What is the base for these characters? Genetic material. Genetic material, there are two types of genetic materials are there. Primitive genetic material is RNA and advanced genetic material is DNA. In this RNA, first formed genetic material, and in this genetic material, now next letter, because of absence of in this DNA, in this RNA, due to absence of 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 right. RNA doesn't have stability. RNA is has RNA has very less stability. RNA has very less stability. So what is the reason for this? So the stability is due to presence of RNA has uracil. So that is one of the reasons for its less stability. RNA has 2' OH group. So these are the two things. We will discuss what is the 2' OH group. So because of these two things, RNA has very less stability. So because of these two, uracil and uracil is present as a genetic material. Sorry, in this uracil is a nitrogen base. Because of this nitrogen base, RNA has very less stability, and it has 2' OH group. So due to presence of this 2' OH group, also it has very less stability. And RNA later, what happens later? RNA is replaced by DNA in higher organisms. RNA is only present in higher organisms, and next one DNA. So DNA, let's see the structure of DNA. So DNA structure is called, let us consider DNA is called as polynucleotide strand. The structure of polynucleotide strand, we will the structure of polynucleotide strand. We will discuss the structure of polynucleotide strand, right? The structure of polynucleotide strand. The structure of polynucleotide strand we will discuss now. So polynucleotide strand DNA. So DNA is having three parts. First one is what is nucleotide? DNA is a polymer of nucleotides. DNA is polymer of nucleotides. DNA is polymer of nucleotides. So what is the nucleotide? A nucleotide contains three things. First one is ribose sugar. A nucleotide contains three parts. First one is ribose sugar, and first one is ribose sugar, and second one is nitrogen base. Second one is nitrogen base, and third one is phosphate moiety. The third one is phosphate group. The third one is phosphate group. So these are totally phosphate group. So these are the three things present in and on ribose, one nucleotide. The nucleotide is made up of these things. First one is ribose sugar. DNA made up of many nucleotides. DNA is a polymer of nucleotides, and it is also known as polynucleotide strand. And it has DNA has several nucleotides, and each nucleotide structure we have to see. The nucleotide structure we have to see. So nucleotide, first one is ribose sugar. Nucleotide contains three parts. One is ribose sugar. Another one is nitrogen base. Third one is phosphate group. Clear? These are first one is, let's see. So in the nucleotide, what is the first one? Let us see. So the first one is in this nucleotide, the first one is ribose sugar. Ribose sugar. So a ribose sugar, if you see, a ribose sugar is a ribose sugar, if you see, so that will be like this. The ribose sugar is like this. OH group. Here, this is one OH group, and this is hydrogen, and this is one OH group, and this is hydrogen, and this is CH2, and this is OH. So totally, a ribose sugar contains four free OH groups. 1, 2, 3, 4. Four free OH groups are present. This is hydrogen, and this is ribose sugar. The formula of ribose sugar is C5H10O5. This is also known as ribose sugar, also known as pentose sugar. We can write ribose sugar or pentose sugar. We can say ribose sugar or pentose sugar. So this is the ribose sugar. So ribose sugar, if you see this ribose sugar, and another one more ribose sugar is, if you see first one, this is first carbon, this is second carbon, this is third carbon. Clear? This is third carbon, and this is fourth carbon, this is fifth carbon. Clear? So this is fifth carbon, and this is first carbon, this is second carbon, this is third carbon. So these are the carbons. Now first carbon this is, second carbon this is, third carbon, fourth carbon, fifth carbon. So that first carbon OH group, this is first carbon. This OH group is what you will say first carbon OH group. This will be replaced by base. This will be replaced by base, and this will be replaced by one phosphate, and this will be replaced by another phosphate which is present at the 5' end of another nucleotide. So these three will be replaced by uh, different things. For example, if you see first carbon OH group is replaced by base, and fifth carbon fifth carbon OH group is replaced by phosphate. So that's why it is called as 5' end. Third carbon OH group is replaced by another phosphate, that is 3' end. So this third carbon OH group is actually, if it is free, then OH group will be left like this only. If it is joins with any other nucleotide, that nucleotide phosphate group will be joined at this place. For example, here the nucleotide ends means here the third place, what will be there? OH group will be present. So that is first ribose sugar. So this ribose sugar, in this, which one is left free, which is not replaced by anything? This is this OH group. So presence of this OH group is what we will say highly reactive. Highly reactive. So because of this reason, this ribose sugar is highly, what we will say, this ribose sugar is due to presence of this ribose sugar. Now first let's see, this ribose sugar is due to presence of this ribose sugar is less stable, right? One OH group, and this is one OH group, second carbon, and this is one OH group, third carbon OH group, and this is fourth carbon and hydrogen. This is the fifth carbon, CH2, and this is OH. Totally here, how many OH groups are there? 4 OH groups are there. In this 4 OH group, first OH group is replaced by these 3 OH groups will be replaced, but this OH group will be left inside. So that this is which carbon? Second carbon. So the 2' OH group is present. 2' OH is present in RNA. If it is present, then it is called as ribose sugar. This is called as ribose sugar. If the 2' OH group is present, then this is called as ribose sugar. 2' OH group is present. This is first carbon, second carbon, this is third carbon, right? This is third carbon, this is fourth carbon, this is fifth carbon. So 2' OH group. And another one more. This is found in RNA. Next, in DNA, what will be there? This is ribose sugar. If you see DNA ribose sugar, so DNA ribose sugar doesn't contain this OH group here. Hydrogen only will be present. And here generally one OH group is present, and here, uh, this hydrogen group is present, OH, and this OH is present here. So clear. Then this is CH2, and this is OH group is present. Clear. So this OH group is present here like this. Here hydrogen, here hydrogen, here hydrogen, here hydrogen. Now if you see this, so here one OH group is present, here one OH group is present. This is first carbon, this third carbon. But here there is no OH group in this case. So 2' here, what we will say, 2' OH group is 2' OH group is absent, we will say. 2' OH group is absent. So that this is called as deoxyribose sugar. Deoxyribose sugar, we will say, right? So this is deoxyribose sugar. Clear? Now, children, you please observe this. This is this ribose sugar is this ribose sugar is normal because of presence of this OH group in this plate. So this OH group is replaced by which one? This OH group is replaced by this is replaced. These three will be replaced. So this will be replaced. So then this will be replaced by which one? this is replaced by base, nitrogen base, and this one will be replaced by phosphate, this OH group, and this OH group is also replaced by another phosphate of another nucleotide. So one OH group is left. Now let us come to this place. For example, this OH group is replaced by this, these three OH groups will be replaced, and this three OH group are also replaced. So when, if you observe this, here this OH group is replaced by here one nitrogen base. Here one nitrogen base will be added, and here in this place one phosphate is added, and in this place also one phosphate is added. So this phosphate is, I told you already, if it is ends here only, then at the end of, for example, two nucleotide if you take, in this end of this phosphate won't be there, third prime and phosphate won't be there, OH group will be there. If it is joined with another thing, then phosphate will be there here. If it is not joined anything, for example, if it is only last, this is only last nucleotide, then it will have then if it is only last one, so then in this place one OH group will be left. So that is concept. If it is joins with any other thing, for example, if it is joins with some other nucleotide, then here in this place what will be there? In this place, if it is joins with some other thing, so then in this place one OH group, oh sorry, one phosphate will be added. One phosphate is added so that this is replaced three prime end. This is fifth carbon, so that this is called as 5' end. Clear? So like this. But if you observe this, here OH group is, if you observe here, so in this place, if you observe, this is one OH group is present, here OH group is absent. So due to absence of this OH group, due to absence of this OH group, it is called as deoxyribose sugar. And this is ribose sugar, this is deoxyribose sugar. So ribose sugar contains 2' OH group, whereas deoxyribose sugar contains, there is 2' OH group is absent in this. So that is the difference between the ribose sugar of DNA and RNA. You can observe this ribose sugar 2' second place, this is second place. In the second place, one OH group is present clearly, and in this one OH group is absent in this. OH group is not there, only hydrogen is there. So due to presence of this OH group, if it is left free, so that is highly reactive. So it is left free OH group means this is highly reactive. So that in this is the ribose nucleic acid in RNA, and this is the ribose nucleic acid in DNA. That is the difference. DNA. So DNA ribose nucleic acid, and this is sorry DNA. So this is ribonucleic acid, and this is only sorry, this is deoxyribonucleic acid. This is ribonucleic acid. That is the difference between ribose sugars of both. Now next we can go for the next concept is, we can go for the next concept is, that is what do we have to say, phosphate. So nitrogen base, let's go for nitrogen bases. So nitrogen bases, if you see, so nitrogen bases, there are three types. Nitrogen bases are two types. What are the nitrogen bases? Nitrogen bases, sorry. Nitrogen bases are two types. What are those nitrogen bases? Nitrogen bases. Nitrogen bases are two types. Nitrogen bases are two types, purines and pyrimidines. First one is purines. First one, purines. And next one is pyrimidines. Purines are double ringed structures, and pyrimidines are single ringed structures. So purines, what are those purines? First, let's see, adenine. So adenine is like this. Adenine is like this, and here this is adenine, right? So adenine is like this. This is adenine. Here nitrogen, hydrogen. This is one nitrogen, and this is N, and this is NH2, and this is N, and now this will be it is. So these are the double bonds here. This is adenine. This is called as adenine. So in this adenine, if you observe here, this is, if you observe, so this will be first, this is second, this is third, this is fourth, this is fifth, this is sixth place, this is seventh place, this is eighth place, this is ninth place. Ninth place, this is right. And next one, if you observe this second one, so this is adenine and this is guanine. Let's see, guanine, let's see. So this is guanine. Guanine is, so guanine is like this, and this is NH2, this is NH2, and this will be NH, and it will be here. And next, so this is N, and this is NH, and this NH. So now this is guanine, adenine, and now the second one is guanine. So this guanine and adenine. So these two are called as purines. These two are called purines, guanine and adenine and guanine. So guanine here also, now if you see, this is first position, here NH is there, right? So this is first position, this is the first position, sorry, NH won't be there, N only will be there. Mm, N only, right? So then this is first position, this is second position, this is third, this is fourth, this is five, this is six, this is seven, eight, and nine positions. So guanine is present like this. Adenine and this is guanine. So adenine and guanine, these two are called as purines. And nextly, let's see pyrimidines. Pyrimidines are generally, so these are pyrimidines. Cytosine, if you see, cytosine is here, and so cytosine is here. Next one is here, it will be cytosine. Next one in this place, NH2, NH2, and this is NH, this is NH, and this is N, and uh, this is N, sorry. This is NH2 and this is double bond O, right? So this is cytosine. So cytosine is like this. So this is cytosine. Cytosine is a pyrimidine, and this is cytosine. This is cytosine. This is cytosine. Clear? That is cytosine. Next one is uracil. Let us see. Uracil is uracil. This is uracil. So uracil is left like this, two, and in this place two, and one, two, three, four, five, six, and next one is this is N, and this is NH and NH. So this is next. Thymine is like this. Thymine is also, so in this place CH3 is present in this thymine, and this is NH, and this is NH, and this is O, and this is O, right? NH. So this is called as cytosine. This is called cytosine, and this is, sorry, this is not cytosine. This is called as, this is called uracil. This is uracil, and this is thymine. So uracil and thymine. So if you observe this uracil and thymine, if you observe this uracil and thymine, here this is first one, second one, third one, fourth one, fifth one. Fifth carbon, either fifth position, there will be no CH3 in this. So here fifth position CH3 is there. So fifth position CH3 is there. So that what we can say, thymine is also called as 5-methyl uracil, we can say. So thymine is also called as 5-methyl uracil. 5-methyl uracil, we can say. So type it is called as 5-methyl uracil, we can say. That is the concept of totally purines and pyrimidines. Purines and pyrimidines. So nitrogen bases, these are the nitrogen bases. So purines are double ring structures and pyrimidines are single ring structures. So purines, these two are purines, and these three are pyrimidines. Purines and pyrimidines. So purines are adenine and guanine, and pyrimidines are cytosine, and next one uracil and thymine. And what is the common pyrimidine between? What is the common pyrimidine between DNA and RNA? That is the question. The common pyrimidine is cytosine. The common pyrimidine between DNA and RNA is cytosine. Next one, if you observe this, here NH is there, right? Here NH. So then here also NH is there. When you take this, for example, ninth position, here NH is there. This is ninth position, NH is there. This is ninth position, NH is there, right? So ninth position NH is there. So this NH, ninth position, and here also ninth position NH is there. Here first position only. This is first position. So they will replace OH groups in the nitrogen bases. with the ninth position. For example, purine, if it is there, and 1 and 9 bond, and pyrimidine means 1 and 1 bond, we can see this. So 1 and 9 bond. So this is one is related to which one? That one is related to nitrogen. This one is related to ribose sugar one. For example, let us see this. So then let us see all these things. Ribose sugar, for example. Now let us take this ribose sugar. This is ribose sugar. This is OH group, and this is hydrogen. This is, I told you already, here what will be there? Based on that, we can say here hydrogen is there means we have to say DNA. If OH is there means we have to say RNA. I told you already. So this is second position. This second position is very, very important. So this is second position. This second position is very, very important. So that here this is OH group. If it is free, then this is what else? CH2, and this is one OH group. So this OH group is replaced by phosphate. This OH group is replaced by phosphate. So that here let us write phosphate. Clear? And that's why fifth carbon OH group is replaced by this, so that this is called as 5' prime. Third carbon OH group is replaced by another phosphate, so that this is called as 3' and this is called 3' and this third carbon OH group is replaced by this third carbon OH group is replaced by, so this is third carbon OH group is replaced by another phosphate of another ribose sugar, right? Another ribose sugar, so that this phosphate is called as third carbon OH group is replaced here, so that this is called as repayment. And in this place, if you see, this is what hydrogen is the second place. So that what we have to write, 2 dash OH group is absent. 2' OH group is 2' OH group is what we would say present. This 2' OH group is absent, we can say, right? 2' OH group is absent, we can say. Is OH group is absent, we can say, so that this is DNA. Now let's see. So this OH group, if let us consider, so then there is, for example, one adenine or guanine, let us take. So this is cytosine I'm taking here. Cytosine I'm taking. So this cytosine is here like this, one O is there, and here NH2 is there. Here NH is there, right? So in this N, first position, so this N and this hydrogen and this OH will be removed in the form of water molecule. So that this bonding is called as glycosidic bond. This bonding is called glycosidic bond. And this is cytosine. In this cytosine, so this is NH. So this is in this cytosine, this is NH generally, right? Okay. Then this OH group is replaced by, and OH group is, and this nitrogen, this is hydrogen, and this hydrogen, and here another this is NH2, and this is cytosine, right? So this cytosine is like this. So cytosine replaces with the first bond, and this is first carbon. So this replacement is called as, or this bonding is called as one and one bonding. This bonding is called as one and one bonding, right? Next, in this, right? The next one is, let's see, the next concept. What else? So adenine always paired with thymine in 2 bonds, and cytosine always paired with guanine in 3 bonds. Here there is a question. Here there is a question. For example, in a DNA, in a double stranded DNA, for example, in a double stranded DNA, in double stranded DNA, or let us write only dsDNA. In dsDNA, double stranded DNA, in a double stranded DNA, adenine is 30 percentage. Adenine is 30% means Cytosine percentage is how much? That is the question. Adenine is 30% means cytosine percentage is how much? So 89%, I told you already, adenine always paired with two hydrogen bonds, cytosine always paired with three hydrogen bonds. Adenine always paired with two hydrogen bonds, right? So that adenine 30%. So this is 30%. 30% means thymine also will be 30% only. Adenine 30% means thymine also 30%. So 30 plus 30 equals to 60%, right? So this is 60%. Thymine plus thymine equals to 60%. And cytosine, totally, cytosine plus guanine will be 40%. Left is 60%. A plus T equals 60 and remaining 40%. 40% is C plus G. C plus G and totally C equals to 20, G equals to 20. That is answer. So, to see cytosine percentage, if adenine is 30%, cytosine is percentage is 20. That is the answer. If adenine percentage is 30 means thymine also will be 30 because adenine always paired with thymine. So adenine percentage is 20, 30 means thymine percentage is also 30. So remaining cytosine is 20% and guanine is 20%. Guanine and cytosine 40% and this is 60%. 30 plus 30, 60%. This is the concept. So this is called as Chargaff rule. This rule is called as, according to Chargaff rule, we will say this answer, right? So that rule name is, what you have to say, the rule name is Erwin-Chargaff rule. So the Erwin-Chargaff rule, right? Erwin-Chargaff rule. So what is the rule of Erwin-Chargaff? A plus G by T plus C equals to 1. The ratio of purines and pyrimidines is always equals to 1. Now let us take the A is how much? A is how much? 30%. Okay. And T is almost 30%. Right? Remaining so 20%. This is 20%. Now let us cancel these things and finally answer will be 1. This is Erwin-Chargaff rule. Clear? Erwin-Chargaff rule. Then this is Erwin-Chargaff rule. That is, Erwin-Chargaff rule is not applicable for which one? Erwin-Chargaff rule will not be applicable for a single stranded DNA. If it is a single stranded DNA, if it is single stranded DNA, it is not applicable to single stranded. For single stranded DNA, if it is Chargaff rule is not applicable. Chargaff rule is not applicable to. Chargaff rule is not applicable. For this, not Chargaff rule is not applicable. Chargaff rule is not applicable for single stranded DNA. Why it is not applicable? Because if two strands are there like this, two strands of DNA is there like this, then we can apply this, right? Two strands of DNA is there means we can apply this, right? Two strands of DNA is there means we can apply because one side adenine is there means another side thymine will be there. This side adenine is there means this side thymine will be there. But if it is single strand DNA means we can't apply this. If it is a single strand DNA, then we can't apply this concept to this. So that single strand DNA, so there will be no chance of this Chargaff rule is not applicable. That's one. Next one is phosphate group. Let's see. Phosphate group. So there are two, there are the phosphate group is added to, for example, let's see. For example, let's take ribose sugar plus ribose sugar plus ribose sugar if you take. So in 2020 NEET question, in 2020 NEET question they have given in ribose sugar, nitrogen base replaces which OH group? In which OH group? Which carbon OH group? Which carbon OH group? Which carbon OH group is replaced by which carbon replaced with carbon? So which carbon OH group? Which carbon OH group is replaced by which carbon OH group is replaced by nitrogen base, replaced by nitrogen base. It is given which carbon replace is which carbon OH group is replaced by nitrogen base. This is the question. Nitrogen base replaces which one? For example, this is 2003 2003 2020 NEET question. So now we have seen already this is ribose sugar and here OH group is there. This is I told you first carbon. Now base replaces this OH group. A nitrogen base will join in this place. If it is a purine, it joins with its ninth position, it is one position. If it is a pyrimine, it joins with the first position. So that purine, if it is purine joins, purine, if purine joins, and one and ninth bonding, if pyrimidine joins, a pyrimidine joins. So then, if a pyrimidine joins, one and one bonding, pyrimidine joins with the first carbon of its position, first position to first carbon, it will join. First carbon OH group it replaces. So that this is 2020 NEET question. And which OH group is replaced by nitrogen base in ribose sugar? This is ribose sugar. This OH group is replaced by this nitrogen base is first carbon OH group. That is the concept. Next, then we can go for the next concept is what is ribose sugar plus? If you take ribose sugar plus, ribose sugar plus, ribose sugar plus nitrogen base is equals to nitrogen base. Ribose sugar plus nitrogen base equals to nucleoside. Nucleoside. Ribose sugar, sorry, ribose sugar plus that is nucleoside. Nucleoside. Ribose sugar plus nitrogen base is equals to nucleoside. Examples for nucleosides. If ribose sugar plus adenine, then it is called as adenosine. Adenosine. Ribose sugar plus adenine, then it is called as adenosine. If ribose sugar plus guanine, so then this is called as guanine. Guanosine. This is called as guanosine. And ribose sugar plus cytosine, cytidine. This is called as cytidine. Ribose sugar plus cytidine. And ribose sugar plus, if it is thymine, then it is called as thymidine. These are thymidine. And ribose sugar plus uracil, uridine. So uridine is generally not found in DNA. Uridine is found in only RNA, but we have discussed that. That's all. So ribose sugar plus nitrogen base is equals to ribose sugar plus nitrogen base is equals to nucleoside. Nucleoside. Ribose sugar plus nitrogen base is equals to nucleoside. And in this ribose sugar plus adenosine is ribose sugar plus adenosine is called as ribose sugar plus adenosine is equals to adenosine. Adenine. Ribose sugar plus adenine is called as adenosine. Ribose sugar plus guanine is called as guanosine. Ribose sugar plus cytosine is called as cytidine. Ribose sugar plus thymine is called as thymidine. Ribose sugar plus uracil is called as uridine. These are all nucleosides. If nucleoside plus phosphate is added, then this is called as, if nucleoside is added with phosphate, in I told you already, and in the nucleoside, how many places phosphate will be added? So phosphate is added at two places. What are the places? Two places. I told you already, ribose sugar. If you see, this is ribose sugar, and this OH group is only, second OH group is only, first OH group is only replaced by base. I told you. This is first carbon OH group is replaced by base. And next one, this is fifth carbon. This is fifth carbon CH2, and here one OH is present. So this OH is replaced by phosphate, or this OH is replaced by phosphate of another 5 prime group of another nucleotide. For example, here another nucleotide is there, means here phosphate will be there. Here another nucleotide is there, means here phosphate will be there. Otherwise, if there is another nucleotide, if there is another nucleotide, then here it will be phosphate will be added. Otherwise, there will be no phosphate in this. So here what will be there? If another nucleotide there, then the real phosphate will be there. This is called as 3-phosphate. And here hydrogen is there, means what you have to say? This is DNA, OH is there, means this is RNA. This is total concept. Right, clear? So this OH group is replaced by phosphate. This OH group is replaced by phosphate. This OH group is here replaced by another phosphate of another nucleotide. If this nucleotide is not there, let us consider. If this nucleotide is not there, in this place, if the nucleotide is not there, then what will be there? Here, only if this nucleotide is absent, if this nucleotide is absent, then what will be there here? Here OH will be left like that. If it is, for example, let us take two nucleotides like this one nucleotide, and this is one nucleotide, and this is one nucleotide, and this is one nucleotide. If you take, means here only phosphate will be there, and the last one OH will be the left in this. So this is 5 prime and this is 3 prime end. But in this nucleotide, if you see here, phosphate will be there in the middle. Here everywhere middle phosphate will be there. This phosphate will be belongs to, for this nucleotide it will be 3rd prime, for this nucleotide this will be 5th prime, like that. So then this is the concept of this is the concept here. Clear? So then nucleotides. So nucleotide plus nucleoside. Phosphate, nucleoside. What is nucleoside? Nitrogen base. So this nitrogen base and this nitrogen base and this ribose sugar together called as nucleoside. So for this nucleoside, phosphate is joined means which place phosphate is joined? At the fifth place, the phosphate will be joined. At the fifth place, phosphate will be joined. So that so this is fifth place. This phosphate is joined at the fifth place. So that this is called as nucleotide. So nucleotide means, so what you have to say, ribose sugar, then nucleotide means acid. The word ends with acid. So nucleotide means this is added with the name is starts with ends with the name ends with the name ends with which one? The name ends with ribose sugar, right? So for example, if you see uh, nucleoside, for example, guanosine plus phosphate. Guanosine is a nucleoside. Guanosine is a nucleoside plus phosphate equals to what we have to say. This is guanylic acid. Guanylic acid. This is guanylic acid. And this is cytidine. For example, cytidine. So, for example, cytidine plus phosphate. So, this is cytidine is a nucleoside plus phosphate group equals to cytidilic acid. Cytidilic, cytidilic acid. Cytidilic acid. Here. So, cytidilic acid. Next one is uridine. Uridine plus phosphate equals to uridylic acid. Uridylic acid. So this is uridylic acid. This is uridylic acid. Uridylic acid, right? So then that is uridylic acid. These are all the names. If it is this guanosine, this is a nucleoside, and if it is a guanylic acid, if it is guanylic acid is there, means then it is called as guanylic acid is a nucleotide. Sorry, nucleotide. These are nucleotides. Nucleotides. So nucleoside names will come as guanosine. Nucleotide name comes as guanylic acid because nucleoside plus phosphate equals to nucleotide. So the cytidine plus phosphate equals to cytidilic acid, and uridine plus phosphate is equals to uridylic acid. And next one is thymidine plus thymidine. So thymidine plus phosphate equals to thymidine plus phosphate equals to thymidilic acid. Thymidine plus phosphate equals to thymidilic acid. Thymidine plus phosphate equals to thymidilic acid. These are the names of nucleotides. So there, totally finally, let's go for the structure of nucleotides. Now we understood one nucleotides are having one nucleotide finally have these are the things. Let's see. So this is ribose sugar plus this is OH group, or sorry, OH group means here what will be replaced? Here OH group is replaced by, sorry, okay, first let's take OH group. First, now let's take OH group. Clear. So now let's take OH group. So this is OH group. This is ribose sugar, and this is OH group. First carbon OH group. This is second carbon. So OH group is there means this is hydrogen. For example, let us take this is then deoxyribose sugar, and this will be another OH. So this will be another OH, and this is CH2, and this is phosphate. This is another OH. So these are totally three parts will be there. So totally four. Sorry, if it is a deoxyribose sugar, three OH groups are present. If it is RNA ribose sugar, then here also one OH group will be present. Four OH groups will be there. But we have taken only deoxyribose sugar, right? Deoxyribose sugar. Deoxyribose sugar. Listen, deoxyribose sugar go ribose sugar go vyathyasa. Let's see. So the difference between deoxyribose sugar and ribose sugar. So in the deoxyribose sugar means here OH group will be there. Sorry, deoxyribose sugar means OH group won't be there. This is OH group won't be there. And if OH group is there means, then what you have to do? If this OH group is there means, now we have to remove this deoxy. Then this will be ribose sugar. Now, for example, if you see this, now this is deoxyribose sugar. Here OH group is absent, right? Here OH group is absent. So that OH group is absent, only hydrogen will be there. So this is deoxyribose sugar. Deoxyribose sugar. In this deoxyribose sugar, this OH group is replaced by which one? Base. This OH group is replaced by which one? Phosphate. And this OH group also replaced by another phosphate of another one. Now, for example, let's take this. This is replaced by nitrogen base. So when it is replaced by nitrogen base, then this is now what you have to say. Ribose sugar already we have taken. Nitrogen base is added means. So this nitrogen base is added means what you have to say. This is nitrogen base is added in this place. Nitrogen base. So when nitrogen base is added, now this is called a nucleoside. And after this, here fifth carbon. This is right. Fifth carbon OH group is replaced by one phosphate continuously. So this is called a 5' end. Next one, if it is, I told you already, if it is not joined with anything, then OH will be left. Then 3' and this is. If it is joined with another phosphate, then what will happen? Here, this phosphate will have this. If, for example, another nucleotide is joined, so then, if for example, here another nucleotide is joined like this, another nucleotide is joined. For example, phosphate is present here, phosphate, and this is called as 3' end. Here OH group is replaced by phosphate, and here OH group is replaced by phosphate. Then when it is joined with another CH2 group, then this is another nucleotide like this. So when another nucleotide is there, then here phosphate will be there. If another nucleotide is not there, and what will be there? This is another base, and this is another hydrogen. Sorry, hydrogen, and this is hydrogen only. And here last OH group will be left in this 3' end. If it is there, it is ends means OH group will be there. If it is not end means here phosphate will be there. That is the difference. Now let us see the structure of our DNA. Let us see the structure of our DNA in this. So then, if you see the structure of DNA, now you see this DNA. DNA structure. This is ribose sugar. This is ribose sugar, and sorry, now this is ribose sugar and this is adenine. Adenine and thymine having two hydrogen bonds here we can see. So here two hydrogen bonds are there. Here two hydrogen, one hydrogen bond, one hydrogen bond. So this is adenine, this is thymine. So next here come to this. Here this is which one? Cytosine plus this is guanine. Cytosine guanine will be in pair, three hydrogen bonds. Cytosine and guanine is having three hydrogen bonds. Adenine and thymine is having two hydrogen bonds. Here this is one polynucleotide strand. This is another polynucleotide strand. This is one polynucleotide strand. One DNA strand it is. One DNA strand it is. Right? One DNA strand. Now, so one polynucleotide strand like this. So now when this here phosphate will be there, when phosphate is present here in this place, the phosphate when it is coming with another nucleotide is there means here phosphate will be there. If another nucleotide is not there, for example, here now you can observe in this place. You can observe in this place. Here last one means here OH group is present, right? Here last one this is last one. Clear? Here it is last one, so that here we can see OH group in this place. When it is, for example, in the middle, for example, if it is middle, then phosphate will be there in this place. That is concept. Next one. Now let's go for packing of DNA. In packing of DNA, so the next concept is packing of DNA. Packing of DNA, you see this. So packing of DNA is here we can see the structures. These are called as nucleosomes. These are called as nucleosomes. This is a nucleosome. Now we can see this is DNA. So DNA is wrapped around 8 histone proteins. DNA is wrapped around 8 histone proteins. Totally, how many histone proteins are there? 1, 2, 3, 4, 5, 6, 7, 8. 8 histone proteins will be there. And these 8 histone proteins are wrapped by two rounds of DNA and forms a nucleosome. A nucleosome contains what are the things? First, if you go for this, so then if you go for this, DNA is right. An histone protein octamer is present like this. An histone protein octamer, right? an histone protein octamer is present like this. Like this. Totally, how many proteins are there? Histone proteins are totally 8 histone proteins are there. DNA is wrapped around this. One histone protein, 2, 3, and 4. Back side also another 4 will be there like this. Right? So totally histone proteins, how many will be there? Totally 8 histone proteins. Around this 8 histone proteins, there will be 2 rounds of DNA will be there like this. 2 rounds of DNA. 2 rounds of DNA will be there. And these 2 rounds of DNA and outside what will be there? This is H1 protein. This H1 protein, this H2A and this H2B and this is H3 and this is H4. So this is totally called as histone. This is totally called as histone protein octamer. This is totally called as histone protein octamer. Histone protein octamer. So around this histone protein octamer, there are 2 wraps of DNA. 2 wraps of DNA is present. The 2 wraps of DNA is, so to DNA contains how many? 200, 200 nucleotides. This is very, very important. Nucleotides. Here in this concept, main this is one important one. In the wrapping of one DNA, 202 strands of DNA, the 2 wraps of DNA, how many nucleotides are present in this? The nucleotide is how many? 200 nucleotides are present in this. 200 nucleotides. 200 nucleotides are present in one wrapping of in a nucleosome. This histone protein octamer, this is histone protein octamer plus 2 wraps of DNA is there, so that this is totally called as a nucleosome. This is totally called a nucleosome. A nucleosome contains how much? DNA. A nucleosome contains DNA which is equals to 200 nucleotides. And histone proteins are generally, a nucleosome contains DNA plus histone proteins. DNA plus histone proteins. How much amount of DNA? In a nucleosome, 200 nucleotides length of DNA is there, which is very, very important. And next one is histone proteins are present. So why histone proteins are used in this place? So this because histone proteins are basic in nature. Histone proteins are basic in nature, and DNA is acidic in nature. So DNA is acidic in nature. This is very, very important. DNA is acidic and histone proteins are basic. Histone proteins are basic. DNA is acidic. Histone proteins are basic. Because of this reason, DNA is associated with histone proteins. Why histone proteins are basic in nature? So histone proteins are basic in nature because they are basic amino acids like arginine and lysine. Arginine and they are arginine and lysine. Arginine and lysine. So these two are arginine and lysine. Arginine and lysine are present. Arginine, histone proteins are arginine and lysine. So histone proteins are made up of arginine and lysine. So that histone proteins are basic in nature. Histone proteins are basic in nature. So because of this reason, sometimes in assertion reason questions they will give one question, that is histone proteins are basic in nature. Why means DNA is acidic. DNA is acidic is not the reason for that. DNA acidic, if DNA acidic means histone proteins are basic, that is not the reason. So why histone proteins are basic means DNA is acidic is not the answer. So histone proteins are basic means they are having basic amino acids like they having basic amino acids like arginine and lysine. So due to presence of this, totally histone proteins are basic in nature. So each histone protein is in the core of histone protein. This is in the core of histone protein. How many histones are there? Totally 8 will be there. So that this is called as histone protein octamer. Totally 8 histone proteins are there. This is one histone protein. So this is one histone protein, and this is one backside also same will be there. This backside like this backside also will be there. So that histone protein octamer. This is histone protein octamer is wrapped by 2 rounds of DNA. Histone protein octamer is wrapped by totally how many rounds of DNA? This is 2 rounds of DNA is wrapped here, right? You can see 2 rounds of DNA is present here. 2 rounds of DNA. 2 rounds of DNA is present. So these 2 rounds of DNA is present like this here. Clear? Are you understanding? two rounds of DNA is present like this, so that this is called as two rounds of DNA is present. So because of this, this is called as histone protein octamer. This histone protein octamer plus two rounds of DNA then forms one nucleosome. And this is called as what is this? This is called as H1 protein. This is called H1 protein, H1 histone protein. The protein which is present outside of the core of histone proteins is called as H1 protein. These are totally important. Listen, important question, children. In this topic, what are the things that are important? Means histone proteins are basic in nature, because in first year biomolecules there is one concept: types of amino acids, acidic amino acids, basic amino acids. So basic amino acids, amino acid, arginine, lysine are basic amino acids. So histone proteins are rich in arginine and lysine, so that histone proteins are basic in nature. As histone proteins are basic in nature and they are associated with DNA, and in the DNA, totally the nucleosomes will be formed. Already we have seen the diagram here. This is the nucleosome. 8 histone protein octamers are there. Totally 8 will be there in this. So 8 histone protein octamers are there. You can see here 8 histone protein octamers. So that this is histone protein octamer plus 2 rounds of DNA is called as one nucleosome. 2 rounds of DNA plus histone protein octamer is called as one nucleosome. Clear? That is totally concept. These nucleosomes, many nucleosomes will form like this. So many nucleosomes will form. Here if you see, one histone protein octamer will form like this. And outside of this, this is linker DNA. This is linker DNA, and this is one nucleosome, this is one nucleosome. Like that, many nucleosomes will form and forms this chromatin. So chromatin coils and supercoils. Coils and supercoil. Coiling means what? Here one time rounds, that is coil, and again the totally supercoiling. So coiling and supercoiling finally forms chromatin, and this chromatin will form finally chromosome. If you see this diagram, you can easily understand what is how the packing of DNA. DNA is double helix, and this is one nucleosome. In this nucleosome, if we see in this nucleosome, each nucleosome contains how much amount of DNA? Each nucleosome contains 200. Each nucleosome contains 200 base pairs of length of DNA present, and these nucleosomes are all joined together and forms coils and forms chromatin. So this chromatin is supercoiled and forms chromatin, and finally this chromatin forms a chromosome like this. So this will be finally forms chromatin. So this is totally the concept of packing of DNA. So if you see the packing of DNA in this way, so this is the best photograph in ever in this. So this is DNA associated with histone proteins, and histone proteins are associated with, and they are joined again, and finally chromosomes will be right clear. So we'll continue in the next class, right? Thank you.